A few years before that was the birth of Jerome Jean Louis Marie Lejeune. Don't ask me to say that again. But this is important because he died on this day 20 years ago, on April 3. And he was a French geneticist who was the first to identify a chromosomal aberration that was linked to disease. He actually identified trisomy 24, and that was causal of Down syndrome. And that was the first thing that really shed light on this idea that chromosomal aberrations can be linked to clinical disorders, which was really not suspected until then. In 1887 was the birth this week of William Cumming Rose. And William Cumming Rose was an American biochemist who did a lot of work on amino acids. Jason, I think you would like this. He discovered, feeding rats, that casein was important, and later on in 36 discovered that threonine was amino acid in casein that was really essential. So then he went back and did multiple studies in rats, changing their diet one by one. A lot of rats, a lot of diets, and he identified that there were 9 essential amino acids: lysine, tryptophan, histidine, phenylalanine, leucine, isoleucine, methionine, valine, and arginine. But what's intriguing to me is that in 1942, Rose was allowed to engage in a study where he urged students, get this, to restrict their diet in multiple ways, to the point that about 10 years later, he came up with the fact that 8 of the 9 original amino acids he describes are truly essential for human health in their diet. And I thought that was intriguing. On this day in 1932 was a publication describing vitamin C. Professor C. Glenn King of University of Pittsburgh described, isolated, identified, and then resynthesized vitamin C, which was very important to curtail the problem of scurvy. But I know that our speaker today has a known friend called James Watson, and he would like to know that this week in 1953 was a description in Nature of the double helix by the paper by Francis Crick and Watson called Molecular Structure of Nucleic Acid, a Structure of Deoxyribose Nucleic Acid. So with that, let me introduce Theodore Segal, who some of you have heard about before. He was not a physician. He was a dentist. And in fact, he obtained his dental degree at U of L in 1954. Then he went on to obtain training in orthodontics at Tufts in 1956, returned to University of Louisville to the dental school in 1969, and was one of 2 founders of the orthodontics department. He was very well known in the academic community. He was considered a great educator and teacher, a humanitarian. He was past president of the Louisville and Kentucky Orthodontic Societies. But he died early, and in 1982, after dying of cancer, his family and friends came together and donated resources to put together this lectureship to medical oncology in that year, 1982. And since then, we've been meeting together to celebrate the work and life of Theodore Segal and learn about the discoveries that are advancing our understanding of cancer. And today we're pleased to introduce one of those speakers who's doing a great job in that area. And for that, I'm going to introduce Dr. Don Miller, Division Director for Medical Oncology and Cancer Center Director, who's going to introduce the speaker of today.
Thanks, Jesse. It's an honor to introduce today's Segal lecturer. Jason Chesney grew up in Minneapolis, St. Paul, did his undergraduate work at the University of Minnesota, during which time he distinguished himself by floating with his mentor down the Mississippi from Minneapolis to New Orleans, and that's quite an accomplishment, and was done, I suspect, as an omen of a successful scientist. Jason went on to get his MD and Ph.D. at the University of Minnesota, went to New York City, where he did his internship and residency at Cornell and at Memorial Sloan Kettering, and then worked as a clinical immunology fellow at the Picard Institute for several years. And then in 2003, we were very fortunate to recruit him out of Manhattan, where he said he would never leave, to Louisville, Kentucky, where he has blossomed as one of our very, very best faculty members. Jason, in an era in which we often say the triple threats are dead, Jason is a true example of a triple threat. He's a great physician, widely regarded as one of the 5 or 6 best melanoma physicians in the country. He has 8 to 12 clinical trials going at any one time. He runs the clinical trials office at the Cancer Center, which manages over 100 open clinical trials and does that extremely well. He's a great teacher, has a very, very active basic science lab, and the goal of any clinical investigator in cancer is to develop a new treatment that makes a difference. And he's going to tell us this morning about PFK 158, which will this month at the University of Louisville enter phase 1 clinical trials, and that's a very, very exciting time. What he won't tell you is that he has another drug close on its heels, and by the time he's 55, we'll have had 2 drugs in patients, both of which are first in their class, first in the world kinds of drugs. And so he's been very, very successful, very well funded. But in addition to that, he's a great academic citizen who has played a major role in helping the Cancer Center get better and helping this department and this medical school get better. So we're honored to have him as our Segal lecturer today.
Thank you. Thank you, Dr. Miller. I didn't know my kayaking trip was on my CV. I didn't remember seeing that, but it was actually 2 separate trips. It wasn't one trip. We weren't that good back then, and it was quite a bit of fun. And someday I'd like to be able to give a talk about those trips. I have a lot of embarrassing pictures of John Eaton without any clothes on in the Mississippi River. So thank you all for coming. I had to ask a lot of you personally to come today because I knew it was going to be a rainy day and it's spring break, and so you can collect your $10 checks next to the coffee table at the end. I'm really privileged to talk about an area that I've been working on since the mid-90s, to tell the truth, and it's an area that we generally call cancer metabolism. And we're working in this area because we realize that there's a huge problem in the United States and frankly globally, and that problem is cancer. And it's expected that we'll have about 600,000 people die this year in the United States of cancer. And the red states on this map are the ground zero for cancer. And you can see right here, here's Kentucky, and I put a number 1 there. And the reason I put a number 1 there is if you go onto the SEER database, we're the top hit in the range, 211 deaths per 100,000. So Kentucky has the most cancer-related deaths in the Union. And this just gives you a sense of the age of the people who die of cancer. And while the median age is pretty high, 72, today's 70 is yesterday's 50. And in fact, if you look, a fairly significant number in the United States, say 14,000 people are dying in their 30s. When you're at the peak of your career, 100,000 people are dying per year of cancer. And so it's a major problem, not just for the old, but also for the young, and it's a major global problem. 8 million people are expected to die of cancer this year alone. And in the time it took me to show you this slide, there's been approximately 15 deaths. So our mission at the Brown Cancer Center is really divided into 3 objectives. Certainly, we're very focused on training the next generation of cancer scientists and physicians, and we do that through graduate students, postdoctoral fellows, residents, interns, medical students, and fellows. We want to provide the most advanced diagnostic and therapeutic modalities there are in the region. That means the best radiation equipment. That means the best physicians, whether they're recruited from within or from out.
Here we go. Okay.
But perhaps most importantly, what we really want to do is reduce cancer-related deaths. To us, the 600,000 is unacceptable. The 100,000 in their 50s is unacceptable. The 10,000 in their 20s is unacceptable. And it's hard in the United States today to have someone come in who's a 25-year-old female and walk in the door with stage 3 melanoma and be able to not say to her, "I can cure you of this," in the Western world. It's a major health problem today. So how do we go about trying to solve the problem of cancer-related deaths? And this is sort of a general schema, but at the Cancer Center, what we do essentially is a lot of laboratory research. Most of the CTRB that we reside in is focused on laboratory research, even though it's called the Clinical and Translational Research Building. We focus on the second floor on drug development, mostly run by John Trent, and then preclinical studies down in the basement. And the goal with all this work is not to get the next program project grant. It's not to get the next R01. It's actually to move our technologies over into the Brown Cancer Center, into the clinical space, and to conduct clinical trials. That's our true goal. Certainly, we want to get grants to be able to support these studies, but our real goal is to move our technologies into the Cancer Center in order to conduct preventative and diagnostic and therapeutic clinical trials. And so I'm going to give you an example today of one of those projects that's going on. We have a lot of really exciting projects, and someday I hope to be able to give you examples of other ones. And so Dr. Roman requires that a speaker that talks at Grand Rounds gives a case report. And I think it's a great idea, and I'm going to give you a case report. This is a 55-year-old male who developed a mole. It was on his neck, and it was there for about 3 or 4 months, and it started to bleed. And so he went to his primary care physician. The primary care physician looked at it and said, that doesn't look like much, but why don't you go to your dermatologist? Go to a dermatologist. And about a month later, he got an appointment with a dermatologist. And the dermatologist first did a shave biopsy, and sure enough, it turned out to be melanoma. And then an excisional biopsy was done, and it was about a 2.5 millimeter deep melanoma. And the patient was referred to the Brown Cancer Center, where initially he was treated by the otolaryngologist. He underwent a wide resection and a cervical lymph node dissection. He didn't receive any adjuvant therapy. He didn't go on a clinical trial or anything like that. And about 6 months later, he shows up, and now he's got a big neck mass. And this is a PET scan that you can see of the patient when he showed up in the clinic. And this was not too long ago. I didn't put the dates because of HIPAA, but this was December 2012 at baseline here. And he not only has a big neck mass, he's also got a liver full of tumors, and his mediastinum has a lot of masses, and a few other spots down here. And PET scans are really useful for an oncologist because you can have an enlarged lymph node, 2-3 centimeter wide lymph node in the short axis, and it can be benign. And you can do a PET scan and find out that because there's no glucose uptake. So the way a PET scan works is we inject fluoral deoxyglucose, F18 labeled fluoral deoxyglucose, into the patient. And about 30 to 60 minutes later, we look to see where the glucose goes. And the glucose is actually being taken up by tumor cells about 10-fold more than normal cells. And when the tumor cells take up the glucose through glucose transporters, they phosphorylate it, and it gets trapped. Normally, a phosphoric glucose 6-phosphate, will go down the glycolytic pathway through the isomerase to fructose 6-phosphate. But because it's deoxyglucose, it cannot be isomerized, and it gets trapped, and it just builds up over time. And what you get when you stick the patient into a big gamma detector are these black spots. So this patient, we told him what was going on, and he said, I have to go home and think about this. Christmas is coming up. Let me come back, and let's just see if these things really are growing. And so he waits a few months, and he comes back, and now he's having trouble walking across the hall. He's got abdominal pain. He's got nausea, vomiting, a little bit of cachexia, and his tumors have clearly gotten worse. And so we've, at this point, gotten the biopsy sent off for a mutation. And in melanoma, about 50% of melanomas have something called a BRAF mutation. RAF is a kinase that's downstream of the RAS oncogene, and it's mutated in about 50% of patients. And we're fortunate in that a new drug came out in 2012 called vemurafenib that's a selective BRAF inhibitor. And so the patient had that mutation and was put on vemurafenib, this BRAF inhibitor, and this is a pretty common response. It's dramatic. The patient knew within a week or two that it was working because his neck mass was going away. And then he started to feel better. And then by the time we rescanned him, he was leading a normal life. He had gone back to work and was doing absolutely wonderfully. And he thought he had been cured. But what's unfortunate about this new technology is it's not like imatinib and CML where you have essentially one genetic alteration and very few others that develop. In this case, you have thousands of genetic alterations, and you have thousands that are developing in real time in the case of melanoma. And by 7 months from presentation, this patient started to show some signs of progression while he was on vemurafenib, the BRAF inhibitor. And then by 9 months, now about 6 months of treatment, it was clear that the drug wasn't working. And even he knew this now because his mass was becoming palpable. And so he was taken off the drug, and he was put on a drug called Yervoy or ipilimumab, which is an immunotherapy. And this neck mass was radiated because it was causing some discomfort. But unfortunately, the tumors grew, and the patient ultimately succumbed to the disease within about a month of this scan. And so This is a very classic case. The median time to progression on vemurafenib is, in fact, 6 months. And it's a major problem in melanoma oncology today because we're so close. We can see the patients getting better, the patients going back to work, their tumors are going away, and then they just start to grow again. And so what's driving all of this? I just showed you all these PET scans, and essentially the PET scan showed us that the guy had tumors, then it got worse, then they responded, then they became resistant and came back. Well, all we're measuring with the PET scan is glucose uptake, as I mentioned. And you kind of have to go back to your biochemistry textbooks a little bit, but the glycolytic pathway is just a series of enzymes that convert glucose down the pathway predominantly to pyruvate, which can then enter the TCA via acetyl-CoA. And so along the way, you're making some ATP and you're making some ribose. And so tumor cells have figured out that this is a good thing, because if you want to grow, you need amino acids, you need lipids that you get off of this, you need nucleotides. And so how did they figure out how to grow? Well, this is a very simplistic version of what we believe is regulating glucose metabolism in cancer today. And one of the big ones is something called HIF-1 alpha. And this is a transcription factor that regulates glycolytic enzymes. And if you look at the purple line, these are all glycolytic enzymes that this transcription factor directly promotes, binds to. And it gets induced by hypoxia. It gets stabilized by hypoxia, but it's also stabilized by the oncogene RAS. And RAS is present in about a third of all human cancers, an activated version of RAS. Downstream of RAS, if you add up all the other mutations in the pathways it regulates, it's probably approaching 100%. In certain types of cancer, like pancreatic cancer, KRAS is mutated in 100% of those patients, whereas in colon, it's mutated in only 50%. That's RAS and HIF-1 alpha are major drivers of the pathway. The oncogene MYC also directly regulates a lot of glycolytic enzymes as well as the glucose transporter. And then a kinase that is now called AKT, it was called protein kinase B for the old folks around here. It directly phosphorylates a class of enzymes that actually allosterically activates a key control point in glycolysis. And you can see this control point, this is being regulated not only by HIF-1 alpha, but also MYC and allosterically by these enzymes. So you could imagine this would be of importance to people who are trying to understand how cancer cells drive glucose metabolism. And so I'm going to just focus on it for a moment. And so here we are again with our glucose going into the cell and down the glycolytic pathway. But here's that control point. And the control point is 6-phosphofructo-1 kinase. We all learned about it in biochemistry. But what you probably don't remember is that it is a metabolic sensor for the cell. The glycolysis gets turned off if you have a lot of ATP because you don't want to waste that glucose. So ATP is a potent allosteric inhibitor of this enzyme PFK1, as is protons that get generated through dehydrogenase steps in the pathway, and then citrate, which is a major metabolic hub in the Krebs cycle as it builds up. So these are all negative feedback for the key regulatory step in glycolysis. The enzyme that I mentioned that's being induced by HIF-1 alpha and activated by RAS and AKT is a class of enzymes, and they're generally called PFKFBs. And this stands for 6-phosphofructo-2 kinase, fructose 2,6-bisphosphotase. And there's 4 family members. And they produce fructose 2,6-bisphosphate, which turns this whole thing on. And the rest of my talk essentially is going to depend on you remembering this slide somehow. So I'm going to just reiterate a couple points. And that is, because otherwise you'll spend the next 30 minutes falling asleep. And so fructose 2,6-bisphosphate allosterically activates this rate-limiting step of glycolysis and turns the whole pathway on. You can think of, whenever I say fructose 2,6-bisphosphate, it's like hitting the gas on glycolysis. That's what essentially it's doing. It's turning the whole pathway on, and it's overriding all the normal negative allosteric inhibitory feedback mechanisms that turn off glycolysis in normal cells. And a key point is that it's regulated by a lot of oncogenes that drive cancer. Any questions? Okay. And so we focused on one of the family members in the mid-90s. I was a graduate student trying to find cytokines. I was working in an immunology lab, and I was doing a screen in macrophages, and I found this gene that was unsequenced that got induced in macrophages, and I cloned it, and it turned out to be this thing that regulated glycolysis. And along the way, I heard that the product of this class of proteins had been found to be high in cancer cells. And so I looked to see if it was high, the protein itself was high in cancer cells, and sure enough it was. And then that led to a big study by immunohistochemistry looking in solid tumors and normal tissues. This is a figure from that study, and you can see here, talk's over.
Try to spell again the phosphocrustate.
Can everyone hear me? Would you like me to keep going, or do you want to find out if we should get out of the building? Keep going. Okay. So this is colon cancer, prostate cancer, breast cancer, lung cancer. And these are all the normal tissues next to it. And you can see you're getting a lot more signal in the tumors than in the normal tissue. So it was overexpressed, and that fits with what I've already told you, which is that these oncogenes stabilize the protein. And in fact, if you look through the literature today, including some papers that are very recent, like 2014, 2013, just about all of the regulators that drive cancer in some way end up activating this protein or increasing the expression of this protein. And that includes the one I mentioned, HIF-1 alpha, the estrogen receptor that drives breast cancer, RAS and BRAF. RAS is mutant in 30 to 40% of all human cancers. And then one I forgot to mention, which is P10. And P10 is a phosphatase. It's actually a tumor suppressor. So in about 70% of human cancers, it gets deleted or mutated, and that drives cancer. And when you lose P10, you get an increase in PFKFB3 protein expression and activity. So it drives the whole process. And about 5 or 10 years ago, here at the University of Louisville, a bunch of studies were done looking to see how this enzyme would be important for human cancer cells. And these were RAS-transformed lung fibroblasts, and they were pulsed with a labeled glucose. And what we did was we knocked out the gene and looked to see what happened to the labeled glucose. And when you do that, you get about a 50% reduction in glycolysis. So if you knock this gene out, you knock down glycolytic flux to lactate. Importantly, when you knock this gene out, you also were unable to grow soft agar colonies, and tumors grew very poorly relative to the wild type. So this is sort of one of the objectives of my lecture today, which is that how do we validate a gene as a target for cancer? We want it to not affect normal tissues and affect cancer tissues, and we want it to be able to block tumor growth. And it appeared in knockout studies that this gene could in fact do that. An important consideration whenever you're talking about targeting glucose metabolism is concerned about this. So as I'm talking, neurons are taking up glucose, making ATP, and doing their business. And so if you're going to inhibit glucose metabolism, you could end up giving patients global strokes, essentially. And so that was a concern for us, and we were fortunate in that Salvador Moncada in London was studying neuronal ischemia and had a hypothesis. He thought that PFKFB3 might be important for protection against ischemia, and what he found instead was that neurons don't express it at all. They turn it off completely. They don't use it as a way to regulate glycolysis. Astrocytes do, but the neurons, and this is a neuronal marker. This is the enzyme. This is a nuclear marker, DAPI, and then merged. And if you had the enzyme being expressed in neurons, you'd see a bunch of orange cells here, but you don't. Whereas in astrocytes, you do, because you get the crossover of the red and the green. So that's a very important finding. So neurons don't express this enzyme. And so this is good, because this is the summary slide. So it doesn't matter if you couldn't hear a word I just said. So this enzyme, again, that turns on glycolysis, glucose metabolism, is regulated by oncogenes. It's required for tumorigenic growth. It's required for glucose uptake and glycolytic flux to lactate. It's overexpressed in human cancers. It's regulated by all these oncogenic signals, and it's not expressed in neurons. So for us, all those are good things if you're thinking about developing drugs against a particular target. And so that's where we headed. And we were fortunate here to have a very strong structural biology program in which John Trent runs it. And John is a computational modelist, but an organic chemist by training, a perfect mix. And without even having the benefit of an NMR structure or X-ray structure, he made a computational model of the enzyme and did a screen and came up with an early hit for us. This drug, 3-pyridinyl, 4-pyridinyl, 2-propanone. We called 3PO for short. It's just easier to say. And so this thing was, in theory, was supposed to bind to the fructose 6-phosphate binding pocket of PFKFP3. And we got some protein together. We made recombinant protein and screened it, and along with about 100 other compounds. And this was the one that seemed to inhibit the enzyme the best. And that's shown here. This is a Lineweaver-Burke plot. This is from a paper published in 2008, so this is old news, but Brian Clem did this study, who's now an assistant professor here. And when all these lines converge on the y-axis, that means that you've got competitive inhibition. We didn't quite make the y-axis, but it was awfully close. It was mostly a competitive inhibitor, a little bit of mixed inhibition. But importantly, when we exposed leukemia cells to the drug, what we saw was a reduction in glucose uptake, fructose 2,6-bisphosphate, and lactate. And that led to a later reduction in ATP and NADH. And I've already told you that RAS drives the expression activity of this thing. Well, if you dump drug onto normal human bronchial epithelial cells collected from bronchs, they do just fine. Maybe a slight effect from the vehicle control here, but really do just fine. They grow, they proliferate, they don't really care. You do the same thing with a RAS transform, so isogenic line where you've introduced telomerase, large T antigen, that inhibits P53 and RB, and then an HRAS oncogene, the cells become exquisitely sensitive to the parent compound. And this is a mouse model. Just about every model that we've used, the drug seems to work, but syngeneic models work the best with an intact immune system. And this is a HER2-new transgenic mouse model of breast cancer. So all that's happened is the HER2-new has been inserted into the genome of the mouse. After about 2 months of life, the mouse develops large breast tumors up and down the belly. And we can measure these with microcalipers as well as with CT imaging. And you can see here that the control group grew nicely, up to about 3 grams. That's the total burden in the animal, and about 10% of body weight. And then the animal that was treated daily had a marked reduction in growth, approximately 10-fold reduction in growth. And so we had this lead compound, but its pharmacokinetics were horrible. It wasn't very potent. It was selective, but it wasn't very potent. And we knew that in order to get this into humans, we were going to have to develop it, get intellectual property for it, and work with a biotech company that had the resources to be able to optimize and test the agent. And we were fortunate that along the time that we had this lead compound, a small biotech company was formed in collaboration with the University of Louisville, here in Louisville, called Advanced Cancer Therapeutics, whose goal was essentially to develop technologies both that were discovered by local investigators as well as external investigators, and really work in collaboration with us. And this is a great example of a collaboration where they licensed the compound and then, with a little bit of John's help, developed synthetic derivatives or concepts for synthetic derivatives, and then sent those concepts to India, where it was a lot less expensive to synthesize derivatives of 3PO. And then our colleagues in India would send the compounds back to us, and we would do the recombinant kinase assays and measure fructose 2,6-bisphosphate and glucose uptake and cell growth in the lab here at the University of Louisville. And then when we found good ones, we tell them about it, and they design new derivatives. And we went through multiple iterations of this, screening about 150 compounds ultimately to come up with a lead clinical candidate. And this was really the one step below the lead clinical candidate. Essentially, it's an identical compound to the one that's going into the humans, except for 3 elements that help with solubility. And it's a fairly simple derivatization. Instead of having a pyridinyl ring, you have a quidinyl ring here. And what this ring did for us that we now know in retrospect is it helped to block the ATP binding domain. So this kinase, it's not a protein kinase. It phosphorylates fructose 6-phosphate. And it has to bind the fructose 6-phosphate and the ATP. And we were blocking the fructose 6-phosphate binding domain pretty well, and that's why we had nice competitive inhibition. But we weren't hitting the ATP binding domain. And so by extending the length of the compound a little bit, we were able to block that portion of the molecule, and that gave us about a 100-fold increase in potency. So we really were able to get a much more potent compound in the setting of this collaboration. And this is published work from last year in Molecular Cancer Therapeutics, but This is a Lewis lung carcinoma, probably the most aggressive tumor we work with in the lab. It's a syngeneic animal. It's black 6 mice. You inject these syngeneic lung carcinoma cells subcutaneously, and within 7 days you've got palpable tumors, and then within about 2 weeks you have to euthanize the animal for fear of causing them discomfort. And you can see what happened. The tumors grew, but if we gave this compound daily intravenously, they did not. We had a slight reduction in body weight with our compound, but really not significant. But what was the most exciting thing for me personally was this model, when you inject the flank of the animal, they get a big tumor, but then the cells leave the mothership and go to the lungs and seed and make little lung tumors. And so when they took out the lung tumors, in the control, all of them had lung tumors, but in the treated group, there were none. And we don't know if that's because we blocked the growth and you had to have a certain size tumor in order to metastasize to the lung, or if it was something inherent about the metastatic process that this enzyme is required for. Importantly, if you look at the tumor, had a reduction in the product, had an increase in apoptosis, and that's measured with this cleaved caspase 3. And then we want to have a pharmacodynamic endpoint, and we're fortunate in that the pathway we're targeting is a pathway that's used to detect and monitor cancer. It's glycolysis. And so we have a micro PET scan down in the basement, CTR, and this is the mouse lying on its back, and this is brown fat that takes up glucose. Right here is the mass on the side of the mouse. That's about a 2-gram Lewis lung carcinoma mass. And what we did was we did a baseline scan on a Monday, and then waited a day because the half-life of F-18 is about 100 minutes, so you got to get rid of it before you do another scan. And then we injected the drug and did another scan. And when we did that, we had basically a whiteout of all the glucose uptake, enough so where we think that we're hitting the target, and it's probably leading to the anti-tumor effect. And we can do this with a lot of models. It works great. So colon, this is an example of colon cancer. This has been optimized with irinotecan. It's the most sensitive model for irinotecan, and you get a nice anti-tumor effect with it. The same holds for this enzyme being inhibited. Didn't do it nearly as well with glioblastoma, and I'm not sure why. It could have something to do with the fact that neurons don't express a lot of PFKFB3, but these cells did. It may be that they don't rely on it as much, but we certainly did not get as great of an effect as we had hoped for with this model. This was a flank model, so it wasn't CNS penetration dependent. And then the KRAS positive pancreatic cancer line, this gemcitabine, when this was tested about a year and a half ago, was the first-line therapy for pancreatic cancer in the United States, but that's changed now, but worked just as well. And so we're pretty excited that we're going to be able to have a compound that works in humans. We did continue the optimization from this step, and so this is the compound that I mentioned, PFK15, and you can see that we had this 100-fold increase in potency. We had an increase in cellular cytotoxicity and anti-metabolic effects, and a marked improvement in pharmacokinetics. So look at the half-life of the original compound, and then a 10-fold increase up to 5 hours. And then by throwing on a couple fluoride groups, we were able to further improve that as well as solubility, such that now the clinical candidate, which is PFK15-8, really has appropriate pharmacokinetic properties to go into patients. And so under FDA guidance, we did IND-enabling toxicity studies, and really tried to target the maximum dose that we thought we could put into humans, and we didn't see any toxicity. And the only thing we did see that we can't explain is salivation. The FDA came back and wanted us to do an exaggerated dose scale, so doses that really solubility would not enable us to do in humans, and we did. Eventually, we were able to kill rats, and the only thing we know, all the histopathology was good. We did see trembling, seizure-like activity in the rats, and that may be what's causing the death, but we're frankly not sure. Nothing cardiac or pulmonary for sure. And so there's a phase 1 trial that's open here today. It opened last week. It's opening at Georgetown University via John Marshall, hopefully in the fall. It's run by Rebecca Redmond here at the University of Louisville. Its main goal is toxicity, of course. Very standard 3x3 design. The inclusion-exclusion criteria are standard for a phase 1 trial. Not allowing CNS tumors because of poor brain penetration. And then diabetics are excluded. I'll tell you why in a second. And it's because of the secondary objectives. So, of course, we want to know about adverse events and PK and anti-tumor activity. And then we're going to try to correlate responses with effects on tumor and PBMC, fructose 2,6-bisphosphate. But perhaps most importantly, we've incorporated into the expansion phase PET scans, just like I did with the mouse, where we do a PET scan on day 1. We start the agent, give the agent for a couple days, and do another PET scan, and then do one at the end of 21 days. And the goal is to see effects on glucose uptake before we see effects on the tumors, and that's part of the reason why we're excluding diabetics. And so let's go back to my patient. And so I mentioned the fact that this patient had a great response, and then his tumors became resistant to the BRAF inhibitor. And so everybody's trying to figure out what's going on, why this is happening. And it turns out, this is a paper that just came out a couple months ago. In melanoma, we're fortunate because about a third of our patients have accessible tumors that we can biopsy easily. And this was a study with about 100 patients where they took BRAF-positive patients who had large tumors, put them on the drug. In this case, this patient's large tumor went away after 3 months. They took a picture anyway. And at the beginning, at baseline, they biopsied. And then when the patient progressed, they biopsied. So now they had 2 tumor samples. And then, of course, they had the germline from PBMCs, et cetera. And they took their two tumor samples, and they did comparison whole exome sequencing. And they looked to see what changed from the tumor that was sensitive to now the tumor that was resistant. And what changed wasn't a big surprise. And it's nicely placed here. This 50% is the MAP kinase. These are mutations in the MAP kinase pathway, the pathway that's downstream of all the tyrosine kinase receptors. And these here are mutations in the PI3 kinase AKT pathway. And these are identical pathways, by the way, that regulate PFKFB3. And so you're seeing mutations that drive these signaling pathways. And in fact, the most common mutation that you see is in BRAF itself. You're getting BRAF amplification. So the target of the drug is being amplified in the genome. You're getting higher protein expression levels, and the cells are just becoming resistant because you can't get enough drug in there to nail the BRAF. And so this really piqued my attention not too long ago, about 3 months ago, when it came out on EPUB. And it's because I had read this paper. And so this is a paper by this guy Hicks. And what they showed was when we do a PET scan on a patient, we can't get another one for 2 months because the insurance company won't cover it, right? Somehow, I don't know how, they managed to do a PET scan after 2 weeks on a patient who was getting vemurafenib, this BRAF inhibitor. And so this was the patient's baseline. This is a big bowling ball-sized liver mass, another one right here, taking up a lot of glucose. All the yellow red is the glucose uptake. Here you have the typical PET scan on your far right. And now after just 2 weeks, he actually already had a response. You can see the tumor's a little bit smaller, but it doesn't look especially necrotic. Looks almost very similar to what it looked like before. This one shrunk as well, but you've got complete lack of glucose uptake. I mean, the nuclear radiologist said the SUV, the fluorodeoxyglucose uptake, was identical in the normal part of the liver and the part where the tumor was. If anything, they said it was reduced in the tumor relative to the normal liver. So what this indicated was that this drug, by inhibiting BRAF, you're inhibiting glucose metabolism first, and then you're causing the effects on the tumor, essentially. So it suggested the combination of lots of mutations in pathways that drive PFKFB3 and glycolysis, and then the fact that when you inhibit BRAF, you block glycolysis, suggested to us that maybe PFKFB3 is involved in this. And we came up with this hypothesis, and it's pretty straightforward. I don't know, I'm sure a lot of you have seen these pathways before, but this is the MAP kinase pathway that we talk about a lot that drives cancer. And here's RAS that gets activated, RAF that gets activated, HIF1 alpha that gets stabilized, and our favorite enzyme, PFKFB3, that makes the accelerator. And then there's the second survival pathway, this thing called PI3 kinase AKT, also mutated. So about 2/3 in melanoma are hitting MAP kinase, and about 20% are hitting this pathway, and they all converged on PFKFB3. And so we came up with this hypothesis and wanted to start working towards testing it. And this work was done by a postdoctoral fellow, Julie O'Neill, in the lab. And all she did was pretty straightforward. She took a BRAF-positive melanoma line and put a couple micromolar of vemurafenib on the line, and then measured HIF-1 alpha, which is the proximal regulator of this enzyme PFKFB3, as well as PFKFB3. And what she saw was after an hour, not a big difference, but after 3 hours with a couple micromolar of vemurafenib, she already saw a reduction in HIF, and that persisted to 7 hours, and it was essentially gone at 24 hours. So it killed the transcription factor that's known to be regulate downstream of BRAF. Not a big surprise for us, never actually been shown, but we know that if you overexpress BRAF, you stabilize HIF-1 alpha. At the same time that happened, you markedly reduced the PFKFB3 expression at the level of protein, also at the level of message, which is the proximal target of HIF-1 alpha. Fructose 2,6-bisphosphate went down by 50% at 3 hours. It's basically a more potent inhibitor of this enzyme than our selective competitive inhibitor of the enzyme, which was, to me, remarkable. Now, of course, this is only in BRAF mutant positive cells using a BRAF inhibitor, so it kind of makes sense. But you could start to believe that, wow, if you've got this effect happening within 3 hours and then 0 at 7 hours, this is probably a very important early effect that's then leading to what we want, which is tumors shrinking, tumors not growing. And in fact, the effect on glycolysis doesn't happen until 24 hours. And the effect, I didn't include it on the slide, I should have, the effect on the cell cycle doesn't start until 24 hours. So this is all happening 20 hours before, and we think it's leading to a cascade of effects, ATP depletion, cell cycle blockade, and apoptosis that ultimately is having the anti-tumor effect. And so what we're going to do is we're going to overexpress this enzyme and see if we can block the cytotoxicity of the BRAF inhibitor vemurafenib. So with all this said, vemurafenib is a real problem because it only works for a few months. Maybe we can increase the potency and block intrinsic resistance to the drug itself using our PFKFB3 inhibitor. And this is just flow cytometry looking at propydium iodide and a Nexin-5. This binds the phosphatidylserine on the surface of cells. And this is a very common assay in the lab to measure apoptotic cells, dead cells, cells that are undergoing programmed cell death. And the main late apoptotic cells are up here in the right upper quadrant. And Julie very purposefully used low concentrations of both drugs, single-digit micromolar concentrations, so that she wouldn't get a lot of apoptosis, because you don't see interactions if you kill all the cells. And so you can see she got a small bump here, maybe 10%, maybe 30% here, but then a marked increase in the number of late apoptotic cells as well as early apoptotic cells. And that's shown nicely here. If you do the inverse, these are the live cells, and then what's left of the live cells. So we saw a synergistic interaction between these two. And that's a sort of a theme that I want to convey during this talk, which is that things that are regulated by oncogenes turn out to be very important if you want to override resistance to inhibitors of those oncogenes. And I'm going to tell you briefly about a second example of that. And that's in the breast cancer. I gave a talk here about a year ago talking about the history of oncology, and the history of anti-estrogens is an incredible history going back in time, centuries. But today what we know is that the estrogen receptor, one of the key targets of it is glucose metabolism, not surprisingly, because what does estrogen do, estradiol do when you add it to an estrogen receptor positive cell? It makes the cell take up glucose and proliferate. That's what you have to do. And when glucose is around, a lot of the same things that turn on PFKFB3 have already been described to regulate glycolysis. And you can see this is what I would call a black box here on the cover of Molecular Endocrinology a couple years ago. They didn't really know. They just knew if they took AKT inhibitors, they could block glycolysis. And so people have been working on this, trying to figure out how this happens. And one thing that came up at Washington University that I thought was really cool, and I thought you would like as well, is there's this guy. He's a nuclear radiologist, Barry Segal, and he's been doing this for years. He's taking breast cancer patients and trying to manipulate glucose metabolism using various estrogen and anti-estrogen approaches. And so this is a breast cancer patient with right axillary lymphadenopathy, a mediastinal mass, and he gave the patient estradiol. It was ER positive, and I know everybody goes, "Wow, that's horrible. You should be brought up before the IRB." But in fact, it was only for a couple days, and it was a fairly low dose of it. And when he did that, you could see there was what he called a flare, metabolic flare. There was a marked increase in glucose uptake, not only in the axillary mass, but also in these mediastinal tumors. And what he discovered, he did this with a lot of people, and what he discovered was the patients who responded in this way were the patients who responded to anti-estrogen therapy. And what that told him was that the glucose metabolism was a key target of anti-estrogen therapy. So all these these were people who had the flare, they responded to anti-estrogen therapy. These are the people who had the flare with the dotted line, they lived longer with the anti-estrogen therapy, suggesting that glucose metabolism was very important. And then I haven't seen early PET scans, but certainly when you give patients who have bad breast cancer, and all the vertebral bodies here are loaded with tumors in this stage 4 breast cancer patient, if you give them anti-estrogen therapy, whether it be aromatase inhibitors or fulvestrant, you will block the glucose uptake. And so it suggested to us that, well, okay, there's all this basic science suggesting that estradiol regulates glucose metabolism. Now we've got human studies showing essentially that it does, because we have our favorite diagnostic modality, FDG PET scans. Let's come up with an idea. And no, we don't usually make these little cute figures when we come up with the idea. We do this after we're ready to publish it. But this was essentially the idea on a paper napkin. And the idea was that estradiol would bind to its receptor, which would then translocate to the nucleus and drive the transcription of PFKFB3, which would then be translated into protein and regulate glucose metabolism. Pretty simple concept that PFKFB3 would be important for glucose metabolism. And so Johannes Imbert Fernandez, who's a senior postdoctoral fellow in the lab now, did this study. And all this is MCF7 cells. They're ER-positive breast cancer cells. And she adds 10 and 100 nanomolar estradiol. And within an hour, you get an induction of the enzyme. And what's really neat is it's almost like an ignition switch. It gets turned on, and then it's turned off. The message is, that is. Well, she tracked it. She confirmed that it was binding to the estrogen receptor was binding to the promoter of PFKFB3 using a fancy biochemical method called chip analysis. But take my word for it, she actually confirmed that the estrogen receptor alpha was binding to the promoter. And then she confirmed that the message was being translated into protein. There's a delay, there's a lag. The message goes up at 1 hour, the protein goes up at 3 hours, and essentially, at least in the 10 nanomolar, stays up for out to 24 hours. The glycolysis itself goes up and stays up, which is really interesting. And I see I forgot to put in the product of the enzyme goes up and comes back down, sort of like what you see here, with the fructose 2-sucrose going up and then glycolysis staying up. So it looked like estradiol was stimulating this enzyme, which then was driving glycolysis. But she took it a step farther, and she did this doing two approaches. She wanted to confirm that glycolysis was dependent on this enzyme, that it wasn't just a correlation, but it was actual mechanism. And she did this using siRNA to PFKFB3, which I'm not showing you, as well as the drug, which I am showing you. And so all she did was take these breast cancer cells, stimulate them with estrogen, and here's the PFKFB3 protein going up. And then she added the drug, and the PFKFB3 protein still went up, which we expect because it doesn't affect expression, it affects activity. She then measured the fructose 2-sucrose, and again, we see this increase in fructose 2-sucrose from estradiol, but then if we add the drug, it blocks it. Not a big surprise to us, because it's a competitive inhibitor. Importantly, if she measures glucose uptake or glycolysis, you get the increase, you add the drug, and it blocks it. So it suggested, just like with the BRAF inhibitor, that a downstream key target of an oncogenic signal was PFKFB3. And so just like the BRAF inhibitor, we said, aha, let's take these breast cancer cells, feed them their estrogen that they normally get, and then give them an anti-estrogen. And this is a drug commonly used in the clinic for stage 4. Its trade name is fulvestrant. It's ICI 18278. This is a pure estrogen receptor antagonist, unlike tamoxifen. And you get maybe a touch bump with apoptosis. Again, these were purposely used 1 and 3 micromolar of 158, and then nanomolar concentration of this anti-estrogen. And then again, a nice synergistic interaction where we get a marked increase in dead cells. Recapitulating what we saw in the melanoma system with a different oncogene, a different cell of origin, but with the same key downstream target, PFKFB3, showing synergy. And I include these mice on these things because I always forget to mention that we're now taking these models into mice. So we've already done one experiment, and it looked pretty good. We had a lot of deaths, not from the therapy, but from the estradiol. It turns out if you give mice too much estradiol, you can actually kill them. But we're seeing some synergy in both in the estrogen-based mouse model, and we're working towards doing the melanoma model right now. And so this is my concluding slide, essentially. And so in cancer, you have an oncogenic signal. It could be RAS, it could be loss of a tumor suppressor. Usually it's a combination of both. Our sort of paradigm is that whatever the oncogenic signals are, you're going to increase the expression and activity of this enzyme, PFKFB3, increase the concentration of its product, fructose 2,6-bisphosphate, and then drive glycolysis, survival, and proliferation. Now, if you come up with an inhibitor like tamoxifen or vemurafenib to inhibit the BRAF, or erlotinib for the EGFR receptor, or imatinib for CML, and the list will go on and on and on, no doubt. What's going to happen is you're going to block all of this. You're going to block PFKFB3, you're going to block glycolysis and proliferation. But then, over time, most patients develop resistance, perhaps with the exception of CML, where the majority don't. And so there's bypass. And it turns out not just in melanoma. In melanoma, it's mostly mutations that we can detect by whole exome sequencing. But certainly in breast cancer, a lot of the bypass mechanisms where the patient still has an ER-positive breast cancer, but they're no longer responsive to anti-estrogen therapy, is due to activation of these same pathways, the MAP kinase pathway and the PI3 kinase AKT pathway, which we know will do this, right? Will stabilize PFKFB3 and cause all these things to happen. And so what we're working on today is inhibiting PFKFB3 in the setting of resistance and seeing if we can block glycolysis and proliferation and cure the patient. And so this is my last slide. These are the folks who really drove this project. And I mentioned Brian, an assistant professor here in medicine and now in biochemistry, John Trent, professor with the Cancer Center in the Department of Medicine, Sucheta Kelang, who did a lot of the initial work with knocking down PFKFB3, who's an assistant professor, hopefully about to be associate, and then the two postdoctoral fellows who worked on melanoma and breast cancer, Julia O'Neill and Ioannis Imbert. Where are we going right now? We're really working hard to come up with genetic markers of sensitivity. We want to be able to select which patients are going to respond to this type of therapy, monotherapy. We have an orally bioavailable derivative that's called PFK112 that was essentially developed in collaboration with Advanced Cancer Therapeutics. We're doing a co-crystal structure with the target PFKFB3 in order to conduct rational design of derivatives. It's orally bioavailable, but its potency isn't adequate. We're trying to come up with pharmacodynamic endpoints. And then, of course, the work I just mentioned, we've moved into the preclinical system with a goal of going phase 1/2s in 2015 and 2016 with this drug. And so I'm going to stop there. Thank you for your kind attention, and I'll take any questions if you have them.
Thanks. Thank you, Jason. So Jason, yesterday I was asked to talk to the medical students. I think it was first year class. And we were talking about translational research. And one of the points that I made to them was that if 30 years from now we're treating patients the same way we treat them today, that it would have been a failure. And we can't advance the way that we do things and how we treat patients without having people engage in translational research that you have described here. So I appreciate you engaging in this. The second thing that I want to point out is that on Thursdays I come here and I tell you about Dr. X who in 1887 did something, or in 1953 the description of a certain paper. And I think it's easy for the audience to look track that these are the culmination of years, often decades, of hard work. And many of these things who end up in nothing.
Right.
And so an investigator can devote a career to these things. And even though they may not culminate in a drug or something that affects the physician, others can take advantage of that data and move into a different direction. So I want to remind the audience about that. So I really appreciate this because this is really cool and it gives visibility to UofL, but more importantly, you're doing what you set out to do, which is to provide something to patients that makes a difference. So I have a question for you.
Okay.
And it's about the strategy of doing a phase 1 trial in all kinds of solid tumors. Is that I would think that you wanted to try first in a certain entity, because I would think that you will have a more likelihood of identifying adverse events that could kill your drug early on instead of this. So tell me about the strategy and the thinking behind how do you move this into the clinical phase?
So today, most phase 1 trials are done in a particular cancer type. And in fact, with a particular genotype in that cancer type. And a great example of that was the BRAF inhibitors. That was, of course, only done in melanoma because predominantly melanoma was the major cancer type that had that mutation. We're starting a new phase 1 trial just in melanoma here. It's the only site in the country with a new immunotherapy because melanomas are most responsive to immunotherapy. So most of the time we try to just target one. It's not like the old days 30 years ago where we were taking cytotoxic agents. This is somewhat unique because we're hitting a protein that is regulated by oncogenes that are present in virtually all types of human cancers. And so the goal here wasn't to start off looking at a specific cancer type, but rather to look specifically at toxicity with an intent then to do expansions in we're going to do phase 1/2s probably as opposed to phase 1s only, expansions combining with drugs to reduce resistance. So it's sort of a different strategy. I think the majority of phase 1s today are really going, narrowing it down, not just into cancer types, but also into certain genotypes.
Questions?
Where do I get my $10?
So when the melanoma comes back, it looks like it's coming back throughout the body, not like this one fold that spread, almost like every cell figure out the way around. So is that physiological rather than?
So your question is, you see that when the melanoma comes back, it's coming back everywhere, and you want to know, is it because they metastasize those locations, or is it because of the mutation?
Well, is it like one event that occurs slowly and those cells spread out, or are many different cells coming?
So if you do whole genome sequencing or exome sequencing of each of those tumors, you're going to find very distinct clones with different sets of mutations. That's what we know. Now, whether you're right, I think when you look at a breast cancer patient, for example, you saw the PET scan. It's usually not so rapid, it's not so widespread in most other cancer types, unlike melanoma. And I think that's related to the fact that a typical melanoma has 30,000 mutations, and so it's really true Darwinian natural selection occurring, and so that's the short answer.
So tell me more, because I was thinking about the issue of resistance in a second. And we are, in essence, promoting the development of mutations. These cells are trying to survive at any cost. Mm-hmm. You hit me in one way, I'm going to develop something else.
Right.
So I'm not sure there's any way around that, but one way is to multiple drugs at the same time,
yep.
No?
Yeah.
Which is what I think you're getting to. But is there something about the theory of resistance of tumors that we're learning about how to push some buttons and not others so that we delay the development of new mutations?
Mm. Yeah, I mean, one approach is certainly the concept of antioxidant and reducing mitochondrial metabolism to reduce some of the mutations that occur from superoxide anion, and people have talked about that. Nothing's made it as far as, I think, clinical, and I'm sure nothing is made as far as preclinical, and I'm sure clinical with that. Our approach, so the problem you just described is the problem that we're trying to address by targeting metabolic enzymes. And so what we're saying is RAS and all of its signaling enzymes are readily mutatable, and we know that. They're in portions of the genome. It doesn't kill the cell when they get mutated. All it does is it causes the flags to go up in a different location to signal for X, Y, and Z. What the enzyme we're working on is very different from that. I'm going to show you why. I'll show you this back to this graph, this picture here. So these things all get mutated up here and up here. But PFKFB3 has never been mutated. It's never been described to be mutated in all the tumors that have been sequenced to date. Same goes for PFK1. There are a couple enzymes in the TCA, in the Krebs cycle, that pick up point mutations that affect their activity, but none in the glycolytic pathway. And so these are highly conserved, highly protected genes that the cancer doesn't have an opportunity to affect, essentially. And so what we think by targeting these metabolic effectors of the oncogenes, of the signaling pathways, where you can literally get hundreds, if not thousands, of mutations that can go around the drug. It's essentially like a waterfall. You've got all the water coming down. You put a cup in. That's my vemurafenib, right? it fills up, it stops the water from dropping right at that spot for a few seconds, and then the water just pours over the edge, just like in a signaling pathway. That's essentially what's happening. But they're all that water is coming down in the same place, and it's to trip on this switch to drive glycolysis. So that's why we're going after that, and we think we're going to have a decent therapeutic index, especially when we start talking about combining it with targeted agents. So I think you had a question?
Yeah. So the theory now is that I think when you have this combination of tumors, maybe it's going to go after the body rather than the patient. And so that's the body for move out for therapy. That's not target.
Right. And so, so we're practicing that today in the clinic. We'll go through the first and second line therapies that have been tested and proven in clinical trials, and then, the patient's still alive and the patient still has tumors. Tumors are still growing, and we're scratching our heads. What do we do next, Dr. Miller? And the idea is to start sequencing not just the oncogenes, but just doing whole genome sequencing and whole exome sequencing. Right now, we're generally going to, I think it's 400 to 500 oncogenes. And essentially, you look at the pathways that get activated, and then you try to get the insurance company to approve drugs in those situations. We've had good luck doing that with insurance companies, surprisingly good luck, and we've had some luck with patient care. There are trials designed today in the United States. There's not one open here to do exactly that. It's never been proven. MD Anderson has a big program for this right now. It's never been proven that selecting from the big cabinet which pathway inhibitor I should use based on sequencing is going to help the patient ultimately. It's never been proven in a randomized clinical trial. But we're practicing it today. The cost of whole exome sequencing is down to a couple $1,000s, which is a drop in the bucket compared to what we're spending on oncology care for most patients. So it's going to become part of the standard of care shortly. Yeah. Well, there's
Dr. Harris is only sitting there.
So the question was, does cancer act differently in diabetic patients? And it's interesting. So what causes the insulin resistance and the diabetes is quite distinct, of course, from what happens in the tumor itself. You would presume that having a higher blood glucose would lead to, the tumor is going to have more glucose, it's going to grow more quickly. But the fact is, it's kind of like, so in the lab, if I give a tumor 2 grams per liter of glucose, the tumor cell line, it's going to grow and do just fine. If I give it 4 grams per liter, it's not going to grow any faster. And why is that? It's because it's getting more than enough, right? So in the human, you've got your liver and your skeletal muscle and insulin and glucagon and great glucose homeostasis, and you're delivering all the glucose the tumor needs typically, more than enough, in fact. And so the answer to your question is it's sort of the opposite that you would anticipate. The high glucose causes advanced glycation end products in proteins in the endothelium. And those patients have poor angiogenesis. That's part of the reason why we have to amputate their legs all the time. And that poor angiogenesis slows tumor growth. And so that's the thought process right now. That's what we believe today. But I do think that patients who get, everybody used to ask, why do our patients get cachectic? And, as soon as you look at those PET scans, you start to understand why they're getting cachectic, because the tumors are starving all the normal organs.
Final question.
There isn't one. Oh, there is.
Understanding all this, it seems like it's really not lethal, which slows growth. So all you're doing is this is a chronic illness, allowing longer time for these patients to develop. Still have to make
Yeah, so if you block proliferation, you don't get mutations, essentially. They don't get carried forward. So that's one answer. The other answer, and I didn't show much of it, but these drugs are very potent pro-apoptotic drugs. I mean, they cause apoptosis in the low micromolar range. And if you look at the tumors, even though they're still growing a little bit, they're full of apoptotic cells. So the jury's still out on whether this is just an anti-growth effect. Most of the data I showed you was with 3PO, the lead compound, and then the second generation compound. We think we're close to getting compounds where we just completely eradicate the cancer cells. But you're right. I mean, we're going to end up with toxicity issues there. And what it comes down to is we need a better drug. We need a drug with better PK. We need a better drug with higher potency. The way that's going to happen is if we get a couple responses in this clinical trial, and then the Pfizers and the BMSs and the Novartises are going to go, we have to start jumping on this bandwagon and making, using our armies to make better PFK-FP3 inhibitors.
So I want to leave you with one message, because I think this audience is sophisticated enough to understand the implications of this work. But I want to leave you with a short story. In my prior institution, there were two scientists who, in the early stages of the HIV epidemic, decided to devote their careers of finding and anti-HIV drugs. And the joke around the institution was that these people would throw anything on top of HIV-infected drugs. So if you left your coffee around and 2 days later they found that cup of coffee, they will go back to the lab and throw it in there. Just a few years ago, that institution sold a drug that was developed by these companies for $500 million to a biotechnology company. And today, the majority of HIV patients are treated with that drug or the. I don't know where you're heading, but I certainly would like the company not to be number one in cancer death.
Right.
And I appreciate your work.
Oh, thank you,
Thank
Jesse.
you. Thank you all.
Great, good stuff. Thank you. Take care.
So do you